Every human genital structure begins as the exact same embryonic tissue. Up to a certain point in fetal development, the biological blueprint for male and female reproductive systems is identical. It is only under the influence of fetal sex hormones that this shared tissue differentiates, splitting into the distinct physiological structures of male and female anatomy. Despite this shared origin, the clinical establishment spent decades treating female sexuality as an afterthought, a derivative of the male reflex, rather than a system worthy of independent study. When researchers William Masters and Virginia Johnson pioneered the modern observational study of sex in the 1960s, they provided the first foundational data on human arousal. But even their early frameworks remained heavily male-centric. Analyzing the female orgasm through the mechanics of male ejaculation resulted in a narrow clinical understanding, one that failed to account for the unique neurological and anatomical pathways discovered in recent decades. This chart reveals a glaring statistical disparity in partnered heterosexual sex. Heterosexual men experience orgasm roughly 95% of the time. For heterosexual women, that success rate drops to 65%. This persistent gap forces a specific biological question. Are female bodies structurally deficient, or is the standard behavioral script failing the machinery? To find the answer, we have to audit the entire human sexual response system, tracking the physical sequence from peripheral nerve endings up the spinal cord, into the brainstem, and through the resulting endocrine cascades. This audit reveals a female orgasmic network with anatomical density and neurological pathways that diverge significantly from the male model. The male anatomical baseline is mechanically straightforward. Penile stimulation relies heavily on the pudendal nerve, with the sensitive glands containing approximately 4,000 nerve fibers. Compare that to the female glands, clitoris. It packs roughly 10,000 nerve fibers into a space the size of a pea, making it the single most densely innervated tissue in the human body, surpassing even the fingertips. What is visible on the surface is just a fraction of the organ. The complete clitoris is an expansive internal structure, reaching 9 to 11 centimeters deep, branching into a central body and two legs called crura. Below the crura sit the vestibular bulbs. During arousal, these two elongated masses of erectile tissue engorge with blood and wrap tightly around the vaginal canal. This anatomy dissolves the old distinction between clitoral and vaginal orgasms. Internal vaginal friction applies pressure directly against these engorged vestibular bulbs and the deep roots of the clitoris itself. The female orgasmic trigger functions as an expansive internal sensory network, requiring diffuse stimulation across this entire complex to reach the threshold of climax. In the male system, the route to the brain is singular. All sensory data from the genitals must travel strictly up the spinal cord to register. The female nervous system possesses a unique anatomical override. Sensory input from the cervix can utilize the vagus nerve, a cranial pathway that completely bypasses the spinal column and wires directly into the brainstem. Deep cervical stimulation sends intense sensory signals straight up this vagal line, entirely circumventing the standard spinal infrastructure. The clinical proof for this bypass is striking. Women with complete spinal cord transections, who have zero lower body sensation, are still documented achieving profound orgasms because this vagal connection remains fully intact. Evolution built a redundant, direct-to-brain sensory failsafe into the female neuroanatomy. The male body has absolutely no structural equivalent. As stimulation reaches the brain, both sexes must cross a critical psychological threshold, the deactivation of the prefrontal cortex. The brain has to physically turn off the regions responsible for anxiety and self-monitoring to achieve climax. When that happens, both men and women experience intense activation in the deep cerebellar vermis and the nucleus accumbens, flooding the brain with dopamine. But PET scans reveal a stark divergence in how the two brains process the peak. In men, there is intense, isolated activation in the periaqueductal gray, a specific region in the midbrain. This spike in male midbrain activity serves a single mechanical purpose. It drives the localized motor reflex of ejaculation. Shift over to the female brain, and that localized midbrain spike is largely absent. Instead, there is dominant, widespread activation across the frontoparietal areas and motor cortices. This broad cortical activation proves that female orgasm operates as a complex, whole-body sensory integration event, not a localized physical reflex. Because it engages so much more neural territory, female orgasm is deeply dependent on psychological safety. Even mild anxiety keeps the prefrontal cortex active, effectively blocking the brain-wide deactivation required to reach climax. While the male brain triggers a localized mechanical reflex, the female brain engages broad cortical regions, requiring sustained, body-wide sensory integration. Once the climax occurs, the timeline diverges immediately. A typical male orgasm lasts between 3 and 10 seconds. Female orgasms average 20 to 35 seconds, often engaging sustained pelvic contractions. This brevity in men is followed by a biological off-switch. The refractory period forces an acute chemical blockade that makes immediate rearousal physiologically impossible. This chart tracks the post-orgasmic chemical sequence. Ejaculation triggers a surge in prolactin, a hormone that commands the release of a peptide called somatostatin, which aggressively suppresses dopamine activity and shuts down arousal. Look at the female hormone curves on the same timeline. Prolactin rises much more gently, allowing estrogen, dopamine, and oxytocin to remain elevated in the system without dropping off a cliff. Escaping that sudden somatostatin surge means the female body encounters no physiological barrier to immediate rearousal. The capacity for multiple, repeated orgasms is built directly into the chemistry. The male system enforces an automatic shutdown protocol to conserve sperm and biological resources. The female system is chemically designed to support persistent, repeatable activation. The evolutionary logic of the male orgasm is incontrovertible. It exists to trigger ejaculation, depositing sperm at the optimal location to guarantee reproduction. Since female climax plays no required mechanical role in conception, evolutionary biologists debate exactly why such an intricate system exists at all. One major framework is the mate choice hypothesis. It proposes that female climax evolved as a selective filter, using intense oxytocin release to reinforce pair bonding with highly compatible partners and physically facilitate sperm retention. A competing theory suggests it is an evolutionary relic, a neuroendocrine reflex preserved from ancestral mammals whose ovulation was physically triggered by copulation. Regardless of its origin, because the female orgasm was decoupled from basic reproductive plumbing over millions of years, evolution freed it to become a highly complex, variable, and neurobiologically rich reward system. Which brings us back to this chart. If the female machinery is structurally complex, why do heterosexual women experience 30% fewer orgasms during partnered sex? The biological deficit theory collapses entirely when we look at the control data. During solo female masturbation, the success rate jumps to match the male baseline. The hardware works perfectly. The demographic proof confirms it. In partnered sex between two women, the female orgasm rate hits 86%, virtually erasing the statistical anomaly. The deficit is mechanical, not biological. Standard penile vaginal intercourse provides continuous, direct friction to the male anatomy, but almost entirely bypasses the external glans clitoris, where those 10,000 nerve fibers converge. Add to that a cultural script that equates sex primarily with penetration. This forces many women to perform gender labor, suppressing their own physical needs to maintain a male-centered routine. That dynamic generates anxiety, which chemically blocks the prefrontal deactivation required to climax. With distinct neural pathways, greater anatomical density, and an absence of a refractory period, the female body possesses a complex orgasmic capacity that is often ignored by the prevailing behavioral script.